And then we sacrificed these rats also, and we found, this is, for the pathologists who are here will be familiar, this is the Mason's trichome staining and TTZ staining, which is for fibrosis and for the infarcted area. And in the fetal cardiac MSC group, there is both a decrease in the fibrosis and a reduction in the infarct size. So that was a nice story. But there was a caveat. The caveat we realized very soon is to be able to get aborted fetuses. It's not going to be, maybe in India it's possible, but in other countries it would probably be impossible to get aborted fetuses. So we decided to look at the amniotic fluid mesenchymal stem cells, because literature started coming in that they have a high differentiation potential. So first we began by comparing the amniotic fluid MSCs with bone marrow MSCs, which is doing the basic work, good growth kinetics. The blue line are the amniotic fluid MSCs. But phenotypically, same markers were present, but a higher proliferative potential. And then we looked at the cardiomyogenic potency of these human amniotic fluid MSCs in comparison to the bone marrow MSCs. And we found a beautiful differentiation of the amniotic fluid MSCs into the cells of the cardiovascular lineage, as well as the expression of the cardiovascular genes in them. And then, as you go down the line, you become better and better. We noticed a paper which said that the c-KIT positive amniotic fluid stem cells, which are CD117 positive, have a cardiogenic predilection. We picked up this cue from this paper and did further work not just on whole amniotic fluid MSCs, but on the c-KIT positive subset of amniotic fluid MSCs. We sorted them out through a FACS sorter. And very interestingly, we found that in comparison to the unfractionated amniotic fluid MSCs, the c-KIT positive population had a higher cardiovascular predilection. In all, the p-value was higher when they were made to differentiate. They had a much higher differentiation potential in comparison to the whole population of amniotic fluid cells. And then we did again the in vivo studies, because before you take them to humans, you have to do preclinical animal studies. And this is what we observed. This is a healthy control. This is an MI-induced heart section. And this is an MI-induced c-KIT positive EFMSCs. So you can see very much reduction in the fibrosis. And then we went one step even further, that instead of using cells, why not use a secretome? Because there is always a reservation about cells now. It's more appropriate to use the secretome. And the greatest effect we found was of the secretome. So one thing became very clear, that the c-KIT positive population, and especially its secretome, is very good in myocardial regeneration. And as I said, that this is the action of mesenchymal stem cells is not through transdifferentiation. It's through production of different paracrine factors, And other than that, the mechanism is there, transdifferentiation. And we found that there was a reduction, when we did the section analysis, there was a reduction in the pro-apoptotic genes, pro-apoptotic proteins in the sections, in the heart sections. That is the BAX BCL2 ratio and the cleave caspase 3 levels. The last one is, this is the secretome, and this is the one before that is the c-KIT positive population. So the most profound with c-KIT positive population and even more profound with the secretome. This encouraged us to go into the renal injury model, and very briefly, we are down the line, so we formed embryoid bodies. As you all know, we can form embryoid bodies from the amniotic fluid. That technology came into the world, we formed them. We could differentiate these amniotic fluid stem cells to form the embryotic bodies and express markers of all the 3 germ layers. So they're almost like embryonic stem cells. And when they expressed, because the urine is urine of the fetus is excreted into the amniotic fluid. So that's how we went into renal regeneration, and we found that these rat amniotic fluid stem cells expressed the progenitor markers, renal progenitor markers, and they could be very nicely differentiated into the renal proximal tubular-like cells. And in vivo, we gave them to a rat model of acute renal failure, which is cisplatin induced, and they produced a beautiful regeneration in this model. Here you can see the histopathology slide. And again, when we looked at the mechanism, because always when you have an observation, you want to see the mechanism of action. It was again through the production of anti-apoptotic factors. Again, the BCL2 Bax ratio, the cleaved caspase levels were decreased. So I will end here. I've given 2 examples. And being a bone marrow transplanter, I would like to I began with a case. I want to end with this case. It's a 28-year-old male who presented, this is during the COVID time. He presented after a COVID infection and was diagnosed to have a severe aplastic anemia. The COVID was coincidental. He had to undergo an emergency HSCT. He engrafted very nicely on day +12, very timely. But then he developed what is one of the most dreaded complications of bone marrow transplantation, which is graft versus host disease. In solid organs, you tend to reject the graft. But in bone marrow transplantation, it's the opposite. You're giving a graft which is teeming with lymphocytes into an immunodeficient, immunocompromised individual whose marrow has been ablated. So the foreign T lymphocytes mount a response against the recipient's antigens, which is known as graft versus host disease. So he developed a horrendous graft versus host disease, with a diarrhea which was occurring 2 times, 3 times an hour. He was pouring out. We gave steroids. We could not achieve a response. COVID time, we did not know what to do. So then we gave him, if you recall, I said that mesenchymal stem cells have a beautiful property of immune silence. So we gave him mesenchymal stem cells from a third party because they are not rejected, and the GVHD regressed. And this patient is alive and doing very well, both the patients. So this shows not only the regenerative capability, but also the immunomodulatory properties of MSCs, which actually is very attractive. Which way is stem cell research growing? This is a publication from the University of Cambridge, which I found just outstanding. A synthetic embryo with brain and beating heart has been grown. To mimic embryogenesis is a very, very difficult, almost impossible task. And the scientists have been able to do it. At least it has a beating heart and it has a brain. So this is the way we are going forward in stem cell research. And what is the future? No doubt it's at the cutting edge of medical research. It has promises for several diseases for which no satisfactory treatment exists till date. Like I said, MI, heart is infarcted, you can't do anything about the dead heart. And we're creating organs for transplantation, that's where we've reached. But I would say that we have miles to go, and we should not lose hope, or rather we should continue our efforts in investigating the therapeutic potential of stem cells in a scientific manner, not make tall claims, but do it within trials. And even if it's a negative data, publish it. And of course, if it's a positive data, it's a bonus. And this thing does not occur in one or two cases. You need a series of patients to document it. So with this I end. Thank you very much for your patience.
Thank you, ma'am, for the wonderful and illustrative oration. Thank you very much once again.